Okay, so looking at this math question, go ahead and grab your nervous system part 5 notes. Okay, so this question asks you to calculate Q10. Your Q10 formula is Q10 equals K2 divided by K1, and then raised to 10 divided by temperature 2 minus temperature 1. Okay, a couple things I want you to know. First of all, you got to know what those K's are. That's the rate. So the second rate divided by the first rate. And then know that the 10 divided by T minus T, that's an exponent. It's not multiplied, it's an exponent. Okay, so you would want to fill in 0.0716 divided by 0.0289, and then raise, so exponent 10 divided by 30.4 minus 20.4, which is 10. Should have got your answer to the nearest hundredth, which was 2.48, I believe. Okay, be careful on your rounding. Remember that the math questions are going to be bubbled in like you would a social security number or an address on the ACT. If you round wrong, it's going to be wrong. Like there's no half credit or anything like that. I know it seems harsh when I mark it wrong, but I'm doing it to help you out. One math question is equivalent to one multiple choice question. So it's like right or wrong. There's no partial credit. Questions on this one? Anybody who missed it got a couple questions? A couple things I saw would be the rate divided backwards. So make sure that you do the second rate divided by the first rate. And then another thing, make sure that's an exponent, not multiplied. No questions? All right. The cube here is 1.5 millimeters on each side. Okay, so the surface area should have been just 13.5, I believe. Oh, going back. So surface area would have been a length times height. So just 1.5 times 1.5, and you should have gotten 13.5. And you can't bubble in the unit, but just in case, that's millimeters squared. On the second one, the volume should be 6 times side. So 6 times 1.5 times 1.5. And that gives you 3.4 millimeters cubed. I did that wrong.
Surface area is the one.
Oh, I did that wrong. Dadgummit. Okay, so the volume is length times width times height. So 1.5 times 1.5 times 1.5. Okay, so one thing I want you to know, the unit isn't going to be, you're not going to be able to bubble in the unit. So that doesn't matter. But if you are asked a math question in the writing, include the unit because you're going to get a point for that. So if there's a math question in the FRQ, like they ask you to determine rate of a graph, then make sure you include the unit, which would be the rise over the run. Whatever the unit is on the y divided by the unit on the x. But on the math questions, the unit doesn't matter. On the FRQ, the unit would matter if there's a math. Make sense? Questions? Anybody? Okay, go ahead and grab those notes for me. Nervous system part 5. Okay, so yesterday we talked all about hearing. And we said that a wave comes in, a sound wave comes in, and it vibrates that eardrum, the tympanic membrane, and that vibrates the hairs. And when the hair bends, this is the important part, so hear me say this. When the hair bends, the nerve is depolarized, and that sends an action potential to the brain. Okay, that's the part you got to know. When the hair bends, it's a mechanicoreceptor. When the hair bends, the nerve is depolarized, and it sends a message to the brain. You got that? And then we talked about balance. Okay, today is all about eyesight and movement. So eyesight and muscle contraction. First things you want to already know. Conformational shape change triggers a transduction pathway. I'm just going to die up here. Y'all just keep sitting there. Leading to a response. Okay, so here's what you want to know. The shape change. When the protein changes shape, it sends the impulse. Signal transduction, it starts the the nerve impulse to the brain. So where it says triggers a transduction pathway leading to response, so that conformational shape change makes transduction happen. Okay, the second thing, and this is what the picture's showing you, actin and myosin is a part of the cytoskeleton. Somebody tell me what you know about actin and myosin. They flex muscles, right? Okay, actin and myosin are proteins in the shape of tubules, protein filaments. Actin is thin and myosin is a little bit thicker. And they're in muscle cells, and they flex. So if you look at this picture, the actin is this thin one, and the myosin is this purple one. And they slide together. So all you needed to already know is that it's a protein filament in the cytoskeleton. Okay, and then this picture you should know oh so well. What is that picture of? Signal transduction, right? Reception, transduction, right here. And then the response. But this one specifically we talked about for the secondary messenger, the IP3. Okay, remember IP3 opened up the channel on the smooth ER, and that released calcium, which was used for what? Muscle contraction. And we're talking about muscle contraction today. But instead of talking about the secondary messenger, we're going to talk about what calcium actually does. So how does calcium work once the nerve impulse tells the muscle to flex? It releases the calcium, which makes the muscle flex. Okay, so any questions on this? All right, eyesight. This is a little video clip, I think, maybe.
Most of the eye cavity is filled with a clear, jelly-like substance.
Let me try that again.
Most of the eye cavity is filled with a clear, jelly-like substance known as vitreous humor. Light from an object enters the eye, passes through the cornea, aqueous humor, lens, and vitreous humor, and is focused on the rear wall of the eye, known as the retina.
Okay, so we're going to talk about the retina today.
Thousands of light-sensitive cells called rods and cones, which capture the object's image. These cells send nerve impulses along the optic nerve to the brain.
Okay, so far, I want you to know that light comes in through the lens and hits the retina. That's that part on the back that you see right here. Hits that part. And then, no, we're not done.
The critical central portion.
Stop that. Okay, so light comes in, hits the retina, and that's what's going to send the impulse to the brain. Okay?
Of the retina is known as the macula lutea. The macula constitutes the region of maximal visual acuity, which is used for activities such as reading, writing, and sewing. Tiny blood vessels nourish the retina and macula. If vessels to the macula are damaged, the ability to read fine print or see small objects can be lost. If the damage remains untreated, vision can become blurred. Eventually, maximal visual acuity is lost completely. An ophthalmologist can use a procedure known as fluorescein angiography to examine the blood vessels and pigment cells within the retina.
Angiography. Blood vessels. See.
Through angiography, blood vessel abnormalities can be located and identified for possible treatment. During an examination, fluorescein dye is injected into the bloodstream. The dye, a water-soluble solution, fills the blood vessels in the back of the eye. Because of its fluorescent quality, the dye glows in response to light. During the examination, the flash of the camera causes the injected dye to glow, making the blood vessels highly visible. An ophthalmologist can study the resulting photographs to make a diagnosis and prescribe treatment.
Okay, so looking at your notes, you see the first one. This is just showing you like a bivalve, like a clam or a jellyfish. They have the most simple eye. So just if you ever see that word, oscal is the most simple eye. Okay, and then a platyhelminthes, they have an eye cup, and it's like an indention. How ours dents out, the eye cup is an indention. And the problem with the platyhelminthes, the little worms, they're flatworms. The problem with them is that sand gets up in their eyes, and most of them are blind because the sand scratches their eyes. But they have an eye, which counts. Okay, compound eye. This is where there's more than one lens. And the animals, like insects, look at this picture. That's the coolest picture ever. They actually see, in all those lenses, they see multiple pictures. So they would see like multiples of me. Wouldn't that be cool? You can see a lot of me. Yeah, right? Okay, so on the compound eye, it says invertebrates such as insects, invertebrates. Multiple pictures, and it's great for detecting movement. So I guess flies, I feel like it helps them see like behind them. You know, if you can't sneak up on a fly and catch it. Okay, a single eye, which is like we have one lens, a single eye lens, is good for seeing depth. So we can tell like Hillary's sitting in front of Josh, which is in front of the cabinet, right? So we can see depth much better. Okay, so here's a picture I want you to look at. What happens is that light comes in. Let's go with purple. Light comes in right here through the pupil. That's the part we see. And light hits the retina, which is that yellow part. Y'all see the yellow part, the retina? So light waves come in, hit the retina, and the retina has on it protein receptors. They're proteins that absorb light, and they are called photoreceptors. Y'all see that word? Photo, light, receptors. So they're proteins that receive light, photoreceptors. I feel like it's like photosystems in plants, but they don't have green pigments like chlorophyll. Y'all remember the photosystems they absorb light and they chlorophyll in them? Well, these absorb light, except they have a pigment called rhodopsin. So they don't have chlorophyll, they have rhodopsin in them. Okay, and that pigment allows us to see. So the light comes in, hits the retina, and the retina there's two different forms of photoreceptors. There's rods, which is this one right here on top, named because of the way it's shaped. And there's cones right here. So light would come in and hit the rod and the cone this direction. Well, so light comes in and hits it that way. And the protein changes shape. So when the light hits the protein, the rod or the cone, it changes shape. Conformational shape change. So when the rod or the cone changes shape, it depolarizes this neuron. It depolarizes that next neuron. So light comes in, hits the rod changes shape, and that depolarizes the neuron and sends a message to the brain. Okay, only thing you definitely want to know about this, you want to know that these are photoreceptors. Rods and cones are photoreceptors. What's a photoreceptor?
Proteins that receive light.
So protein that receives light, right? He said it perfectly. It's a photoreceptor. It's a protein that receives light. Okay, they are the most abundant in animals having these structures. So in other words, we have way more rods than we do cones. There are more rods than there are cones. And then they possess a pigment called rhodopsin. It is a combination. It's a pigment. It's going to allow you to see in rods black and white. So rods let you see black and white, and then cones let you see color. Do y'all see under rods the little eye? A shape change allows depolarization. Most important part. Light comes in, it changes the shape of the rod, and that depolarizes the nerve. What does depolarize mean?
Can you repeat that again?
Sodium goes in. Right, so Logan, can you write this? Okay, so light comes in, hits the rod changes shape, and that depolarizes the neuron. You got it? Light comes in, it changes shape, and that depolarizes the neuron. Just like yesterday, when the hair bent, it did what to the neuron? Depolarizes it. And what does it mean to depolarize? Yes, it means sodium comes in, but what does that mean for the neuron? The nerve impulse can go down it, right? So light comes in, the photoreceptor changes shape, the nerve is depolarized, or better yet, sodium comes in, and then the impulse is sent to the brain. Okay, so it says on cones, this is a photoreceptor, they're outnumbered. We have way less cones than we do rods. So we see black and white and shades of gray better than we do color.
So dogs don't have cones? Is that why they see black and white?
Yeah, let me get to that in just a second. So they are found in vertebrates, but not all. So y'all see where it says they're found in vertebrates, but not all. I know some dogs are colorblind. I don't think all dogs are, though, are they? Oh, you think all dogs are? Okay, I was thinking some of them were. And it's because they don't, like DNA makes RNA, makes protein. Well, they don't have the DNA to make that protein. So just like someone with diabetes doesn't make insulin, someone who's colorblind doesn't make cones. Right? I know deer don't see color also. Like, so when hunters are looking for camouflage, they're not looking at the color of the camouflage. They're looking at the pattern of the camouflage. Or at least that's what they should be looking at. You know, like the leaf and the stick, like how it looks on the shirt or the jacket or whatever.
And orange.
The orange is not for the deer. The orange is for so the person doesn't shoot them. The other people in the woods. And I think it's the law. You have to have on like X amount of orange.
Has to be a square foot.
A square foot. Are you a hunter? Did you know that deers couldn't see color? Oh, okay.
People that are red, green colorblind, do they only have like some?
They only have some of the kinds, right? Mm-hmm. Okay, and then the last thing on cones, it says they possess photopsin pigments, which are red, blue, and green. Okay, raise your hand if you know somebody that's colorblind. Tell me who it is. Drew. Evan. Anybody else? Who?
My brother.
Brother.
And his dad.
Dad.
Was our coach.
Coach. What's his name?
His name's AJ.
Okay.
My dad.
Dad. Anybody else?
Zach Miso is our
Anybody else? Anybody else? Anybody else? All right. Well, what did you notice about all those people?
They're all boys.
They're all boys. Now, some girls can be colorblind. Normally, there's like one person that knows a girl. Okay. The gene to make the cone is on the X chromosome. So, girls, we got 2 X's on our 23rd chromosome. So, everybody has X, 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 X. Girls have X, X at the end. Boys have X, Y at the end. Right? So, boys only have 1 Y. They have all X's and 1 Y. So, on that 23rd pair, girls have X, X, boys have X, Y. Well, the gene to make cones is on the X. So, girls have 2 X's, where boys only have 1. So, if I had a mutated X, to make me colorblind, or I didn't have the gene to make the cone, I have a backup plan. Girls have 2 X's. So, okay, questions on this. Rods and cones, conformational shape change, depolarization, nerve impulse sent. Okay, I have some more videos. Aren't you excited?
Yes.
Oh, that's how I remember that. Orange cones. Cones in the road are orange. They see color. Rods black and white, cones color. Okay, so this picture just shows you, if you can see it, it shows you the neurons right here on this side and the cones and rods on this side. So this one is a cone and that one is a rod. Right here, this one's a cone, that one's a rod. Okay, so light comes in, hits the rods and cones, and then it's sent to the brain. Watch again. Light comes in, sent to the rods and cones, sent to the brain. I just thought that was kind of cute.
Photosensitivity in animals depends on a family of pigments called rhodopsins.
Rhodopsins.
In the human eye, light must travel through several layers of neural cells before it can be absorbed by the rhodopsin in rods and cones. Rods and cones are also called photoreceptors. The rod cell, named for its shape, is a modified neuron that does not produce action potentials, but it does release neurotransmitter from its base, where it forms synapses with the next neurons in the visual pathway.
Okay, so that's what I wanted you to hear that. The light comes in, hits the rods and cones. The rods and cones release a neurotransmitter. So the rods and cones release a neurotransmitter that causes an action potential. So I'm going to rewind it a little bit.
But it does release neurotransmitter from its base, where it forms synapses with the next neurons in the visual pathway.
So it releases it from the base.
Each rod has an outer segment packed with membranous discs. The disc membranes are stacked inside the rod cell outer membrane. In addition to rhodopsin molecules, the disc membranes carry G proteins, also called transducin, and molecules of the enzyme phosphodiesterase. The rod cell outer membrane contains a type of sodium channel that is held open by the binding of cyclic GMP molecules. The open sodium ion channels allow sodium ions to flow spontaneously from a higher sodium
So that's just depolarization. Sodium floods in.
cell to a lower concentration inside the cell, thereby depolarizing the cell.
Sodium comes in, that depolarizes the cell.
Light sensitivity begins with the absorption of light by rhodopsin. Rhodopsin consists of a protein part called opsin and a light-absorbing part called 11-cis-retinal, which is
So this
greater
is
than the center
inside
of
the
opsin and bound
photorhodopsin.
covalently to it. When 11-cis-retinal absorbs a photon of light,
Oh,
to it. When 11-cis-retinal absorbs a photon of light energy, the molecule rotates around an internal bond, straightening out, and becomes all trans-retinal. The shift in retinal forces a conformation change in the opsin.
Conformation change.
Protection of light. The photoexcited rhodopsin activates transducin by stimulating it to release GDP and pick up GTP.
GDP, that's just energy, guanine triphosphate.
Phosphodiesterase. Activated phosphodiesterase then converts cyclic GMP to GMP.
Cyclic GMP is just like cyclic AMP, but with a G.
Allows
Guanine instead of adenine.
to close. Pumps in the cell membrane continuously work to drive sodium ions out of the cell, so the cell can now become more negative. The membrane hyperpolarizes. Signal transduction cascades have enormous amplification abilities. In this example, each molecule of photoexcited rhodopsin can activate several hundred transducin molecules, thus activating a large number of phosphodiesterase molecules. The catalytic capacity of phosphodiesterase is great. One molecule can hydrolyze more than 4,000 molecules of cyclic GMP per second. Thus, a single photon of light can cause a huge number of sodium channels to close. To test how a rod cell responds to light, we can penetrate a single rod cell with an electrode and record its membrane potential in the dark and in the light. In the dark, the membrane potential is around minus 35 millivolts. This value is considered depolarized, that is, relatively close to zero. In the dark, when the cell is depolarized, it continually trickles out neurotransmitter from its synaptic terminals. A brief light stimulus triggers the membrane to get more negative, that is, to hyperpolarize. As the membrane hyperpolarizes, the cell releases less neurotransmitter onto the next neurons in the visual pathway. A brighter light stimulus results in an even stronger hyperpolarization and an even greater reduction in neurotransmitter release. Note that this is the opposite of what other types of sensory receptors do. They typically depolarize and release neurotransmitters in response to a stimulus. To be a system sensitive to light changes, the signal transduction cascade needs to turn off soon after the signal has stopped. With vertebrate eyes, the retinal and the opsin separate from each other soon after light absorption, causing the molecule to lose its photosensitivity. A series of enzymatic reactions is then required to return the all-trans retinal to the 11-cis isomer, which then recombines with opsin so that it once again becomes the photosensitive pigment rhodopsin. Additionally, the G protein has an automatic shutoff mechanism. After a short while, it cleaves its GTP to form GDP and thereby becomes inactive again. Without the activated G protein, the phosphodiesterase stops its activity, while another enzyme in the cell replenishes the supplies of cyclic GMP.
Secondary
the sodium
messengers.
channels open again, bringing the cell to its resting state.
Okay. So this I just thought was interesting. It's not anywhere in your notes. But people who are nearsighted, they have spherical lens. So the lens of their eye, this part right here, the lens right here, is more rounded. And that's why they're nearsighted, because their lens is a different shape. Okay, and then people who have distant, like farsighted, is because theirs is flattened. I've actually read it in books that it's like a football shape instead of being like ours.
Can you like look at it and tell, or is it some kind of like,
Say that again?
Can you like look at it and tell if it's circle, or is it some kind of
Oh no, it has to be a machine. Like you and I can't look at it, but an ophthalmologist can with their little special glasses. Isn't that neat though? Okay, so are any questions about vision before we move on to locomotion? Questions? Y'all, do you understand light changes the shape of the rods and cones and that depolarizes it? Okay, and also note photorhodopsin. That word is important. Okay, so movement. In movement, there's two things working against you: gravity and friction. Okay, so two things working against gravity and friction. It says overcoming the force of gravity and overcoming the force of friction. So if you're a fish or you're an aquatic animal, swimming, there's no gravity, but there's a lot of friction. Right? That's why swimming is hard. So there's not much gravity because the buoyancy of the water, but there's a lot of friction. So how do they overcome that? With their little torpedo bodies. Right? So it says water, there's little gravity to overcome, but much friction, having a fusiform. So they overcome that with the adaptation of that shape. Their torpedo shape, or fusiform, that word. You like the torpedo? Me too. Okay, so land animals, we don't have much friction, right? It's not hard to walk through the air, but we have lots of gravity pulling us down. So in order to overcome the gravity pulling us down, we have muscular limbs, like our legs and rear end, to help us stand upright against the gravity. So we have muscular limbs to overcome our problem. Our opposition is gravity. Questions? This is kind of common sense, though. And then birds, or organisms that are flying, they got both. They have gravity, but then to fly through the air, there's lots of friction. So how do they overcome it?
a torpedo, but they have legs?
Do what? They have hollow bones, so really light, and their feathers are really light. So flying has gravity and friction. Flying's hard, that's why we can't do it. Even if you had wings. Okay, but so in order to overcome that, like they have wings, which is a shape, like they have light feathers and light bones to overcome their problems. So fish have friction, so they have a fusiform. We have muscular bone or muscular limbs on our legs to overcome gravity. And then birds are just lightweight, so they can fight against gravity and friction. Questions on those? All right, muscle contraction. I put this in there. It's not in your notes, but this is just from biggest to smallest, in case you ever see these words. So a muscle is all the fiber together. Like your bicep is 2 muscles. Bi means 2, right? You know that from your prefix and suffix game. Bi means 2. So the whole muscle. Okay, well, muscles are made up of fibers. So there's fibers in the muscle. And there's like one fiber is really a bunch of muscle cells. Okay, and then there's the cells in the fiber. And then myofibril is just a portion of the fiber. So like inside the fiber, there's like a smaller portion called a myofibril. So inside the cell, there's myofibrils. And then there's sacromeres. That is a portion of the cell that's got the actin and myosin in it. I'm going to show you a picture. I can go ahead and show you a picture right here. This is the sacromere. Like this is a muscle cell right here. So this portion right here is the sacromere. So that's inside a muscle cell, a portion. It's got the myosin, which is thick bands. I believe it was purple on that first picture we looked at. And then the actin, which is a thinner band. So if you want to write those down, they're not in your notes. So The only one that you probably don't know is sarcomere. So note that one. If you ever see that word, just think muscle, so you can answer the question. Oh, going back. No, you're fine. Okay, look at this picture. What do you notice is the difference between the relaxed and the contracted sarcomere? So y'all check that out. Do what now? On which one? Yeah, they're okay. They're not as close together on the middle one as they are on the bottom, right? So they're not as close together. What else do you notice?
There's like a white. There's more white in the relaxed.
Yeah, you can see the white area right here, and it's not right there. Okay, one thing I noticed, you see how the actin, like the yellow lines right here, aren't touching? But over here, they're like crossing. Can y'all see that? See how what they're doing is they're sliding together. So the actin and myosin, they're like this when the muscle is relaxed. And then they slide together and the muscle flexes. So that's called the sliding filament theory. The actin and myosin slide together. Think about it. When the muscle's like this, when the muscle is relaxed, it's soft, right? It's not dense. And then when you flex your muscle, the muscle gets shorter. So see my hands get shorter. And it gets more dense. It's not as flexible. It's not as soft. It's hard. Does that make sense? So on this one, you can tell on the top one, if you look right here, you can tell the actin and myosin haven't slid together. And then you see they get closer. And now do you see how like the purple touches from Z-band to Z-band? And the yellow, it's touching in the center.
Why is it a theory not
I don't know. I guess because it's so small we can't see it. That's a good question. She said, why is it a theory, not a law? I guess because we're just assuming. No, not assuming. But it's just so small. Like this is way inside a muscle cell. Okay, so the actin is the yellow one. And the myosin is the big one. And they put together. Okay. Remember, I got this video for you.
Skeletal muscle is composed of bundles of elongated muscle cells called fibers. Each muscle fiber or cell contains thread-like structures called myofibrils. A myofibril is made of units called sarcomeres, joined end-to-end by Z-lines. Each sarcomere contains both thin and thick strands of proteins known as myofilaments.
So that's a sarcomere right there. It's got
The thin myofilaments are attached to the Z-lines at both ends. Thick filaments are grouped in the sarcomere center. When thin and thick filaments interact, they slide past one another. This sliding action pulls the Z-lines toward the center, and the sarcomere shortens. As the sarcomeres shorten, the entire muscle shortens or contracts.
Oh, no. Okay, so one thing I just wanted to add in right here. Muscles always pull. We were way into that, huh? Okay, muscles always pull. They never push. So if you think about for just a second, you all know where your bicep is, right? Okay, so when I flex my bicep, it's going to, think about it, the actin and myosin are sliding together, right? So now my muscle is more dense and it's smaller. Smaller this way in length. Okay, so my muscle right here pulled my arm up. So your bicep is attached right here at this joint and right here at this joint. So when you flex your bicep, it pulls your arm up. Okay, so will my bicep push my arm down? Can the actin and myosin push my arm back down? No, not at all. Well, Holly, what do you think? Can it push it back down? No, it can't, if you pay attention. The tricep right here, underneath your arm, when it shortens, it straightens the arm. So this one shortens and pulls the arm, while this one shortens and straightens the arm. Got it? So if you wanted to bring, now look at my neck. If I wanted to bring my chin down, Is it the muscles in the back that push my chin down? Or is it the muscles in the front that pull my chin down? The muscles in the front. Okay? If I want to bring my head to this side, it's got to be the muscles on this side of my head that pull it down. If I want to bring my head back right, then this side pulls it up. Right? Okay, think about your calf muscle. When you shorten your calf muscle, what happens? Your heel comes up, right? So think about if you were sitting on your bottom in like a pike position, but you can think about standing. So when you shorten your calf muscles in your back, your gastrocnemius, when you flex that, your heel comes up. Now you know your calf muscle is flexed, right? Okay, now in order to straighten it, the muscles on the front have got to pull it back down. Your shin muscles. Got it? Sort of? Yeah, that makes sense? Okay, so back to this. So y'all know that it called these Z bands right here. Actin is the thin one, myosin is the thick one. Okay.
Thick myofilaments are made of the protein myosin. Individual myosin molecules have a two-part head and a long tail.
Head.
A myofilament may contain 200 or more myosin molecules bundled together. The tails form the center of the bundle. The globular heads project outward and in opposite directions at each end. These projections, called cross bridges, have hinges that allow them to bend during muscle contraction. Thin myofilaments contain 3 different proteins. Actin, the primary protein component, has 2 rows of molecules coiled into a helix. The second protein is tropomyosin. These long, narrow molecules wind along the grooves between the actin. The third protein, troponin, is a complex of 3 subunits. Troponin is bound to tropomyosin at regular intervals along the groove.
Okay, so let's go over what this is real quick. The yellow, like beaded looking ones, that's actin. That's the thin protein filament, right? Okay, and then there's this thread thing over it called tropomyosin. Do y'all see that? Emily, do you see that? Okay, and then there's another protein that's attached to it. So, okay, I told you that the actin and myosin slide together, right? The tropomyosin thread is blocking the myosin's active site. So, where the myosin wants to bind, let me say that again. Where the myosin wants to bind on this actin, this thread is blocking it. It can't bind when the thread is in the way. Okay, so what happens is when calcium binds to actin, it moves this thread
Molecules toward the center of the sarcomere. This pulling action is called a power stroke.
Okay, so look at what's happened right here. The first thing that happened was calcium binded. And that moved this whole unit right here. The tropomyosin thread moved. So now the myosin can bind. So that tropomyosin was blocking where the myosin wanted to bind. So once the calcium binds and this part moves, now the myosin can bind. Does that make sense? Okay.
For contraction to continue, ATP must bind to the myosin cross bridges so they can detach from actin. The splitting of ATP energizes the myosin cross bridge, thus preparing it for another power stroke.
So the splitting of ATP caused it to relax. The splitting of the ATP caused the muscle to relax.
Each power stroke pulls the actin closer and closer to the center of the sarcomere. As the myofilaments slide, they continue shortening the sarcomere and the entire muscle contracts. Contraction ends when nerve impulses cease. Calcium is actively transported back into the sarcoplasmic reticulum away from the myofibrils. In the myofilament.
Okay, so looking at your notes with me now, it says, first of all, muscles pull. So highlight pull. Okay, so step 1, the neurotransmitter acetylcholine is sent. So looking at the picture right there is acetylcholine. Look at that picture. What's happening? Acetylcholine does what? Attaches to the protein receptors, right? So on step 1, it said the neurotransmitter acetylcholine attaches to the receptor proteins. Okay, step 2, you know this story already. Acetylcholine attaches, and what goes inside? Sodium. Sodium floods in. So look at your notes. It says depolarization. Sodium floods in. Okay, step 3, it travels. So now the sodium, or the action potential, is traveling right here in to the muscle. So the sodium is now in the muscle, down the T-tubule. The action potential travels down into the cell. Okay, step 5 says the sarcoplasmic reticulum, which is all this right here, the action potential travels. So the action potential travels down into it. So it travels along 3 and 4 could be one step. Don't y'all think? Down the tubule, down into the tubule more. Okay, so right here, this is the smooth ER. It's called sarcoplasmic reticulum. But if you wrote smooth ER on a test, you get the point. Okay, it releases what? You know this story. Calcium, right? So now calcium comes out. Where does calcium bind? To the actin. Okay, so it says calcium. On number 5, it says calcium comes out of the sarcoplasmic reticulum. On 6, it says the calcium ions bind with the troponium complex. That's that little thread and that little three-part protein that you saw on actin. So it binds right there. Do you see that green dot right there inside that purple circle? So calcium binds. It rolls over. Does it say rolls over? Rolls off. So that thread rolls out of the way. And when it does, it opens up the binding site for myosin. So now that calcium is bound right there, look at my picture. Calcium binds right there. It opens up the spot. Now myosin, which is right here, can bind. See how it binds? Let me erase my marks. So there's myosin right there binding. And then they can slide together. ATP is broken off and it relaxes. Okay, look at this picture. So this shows you calcium right here. Oh, let me go back. This shows you calcium right here binding. And it rolls that over so that myosin, see this right here? So that myosin with its little myosin heads can attach to actin and slide together. Okay, one thing I just thought was interesting. What is the difference in making energy with oxygen and without oxygen? In our bodies. Without oxygen, you only do glycolysis. Good. And what else? And then with oxygen, you can do all 3 steps, right? So you make more energy with all 3 steps. And without oxygen, you make what as a byproduct? Lactic acid. Okay, so without oxygen, you don't make as much ATP. Without as much ATP, you cramp up. So if this is just me thinking, it takes that ATP to relax the muscle. So if you don't have the ATP, it's like a Charlie horse cramp or like a cramp in your side. But a lot of times that's from moisture. You don't have enough liquid either. Okay, questions. Questions on that. Look at step 8. It says ATP is used to phosphorylate the myosin head. Just know that this requires energy. That's all you need to know. And you want to know this is called sliding filament theory. Okay, and then one more thing I want to make note of. On it says muscle relaxation. I used the word acetylcholinesterase this time instead of just cholinesterase. What does acetylcholinesterase do?
Closes the...
Good. It breaks down acetylcholine, which would close the gate. It breaks down acetylcholine, which would close the gate and stop the muscle from flexing again. Stop. If acetylcholine's not there, the calcium's not going to be released. The calcium's not going to bind to the actin. The myosin's not going to bind. It's not going to flex. Got it? Questions? Get out sheet of paper. No, use the back of your math quiz. Sorry. Back of your math quiz. Back of your math quiz. Put your notes away, please. Okay, so in order to get the 3 points, you've got to say there's gravity, there's friction, and then the anatomical feature. So, let's say you chose, I'll go through all 3 of them. Let's say you chose the fish. Then there's not much gravity. There is a lot of friction, and they have the fusiform body. Okay, let's say you chose the land animal. There's a lot of friction. There's a lot of gravity, and they have hollow bones. Let's see. Oh, that was a flying, the flying thing. Okay, the land animal, they would say there's not much friction, there's a lot of gravity, and we have the muscular limbs. So to get 3 points, you had to say something about gravity, something about friction, and something about the body. Turn it in right here when you're done.